Biological inputs are one of the most talked about topics in modern agronomy and one of the most rapidly evolving segments within the ag industry. Walk through an ag retail store today and you'll find an entire section of the shelf dedicated to products that weren't part of most crop plans even a decade ago. Products built around living organisms, natural extracts, and biological processes. The biological inputs category has grown from a specialty corner of the market into one of the fastest expanding segments in all of agriculture. And the growers and agronomists adopting these products aren't doing it on faith. They're doing it because the results are beginning to show up in their programs. But growth and clarity don't always move together. The biological input space remains one of the more complex corners of the industry to navigate. The terminology overlaps, the label language is inconsistent, and the question of how these products actually fit alongside or within a nutrient management program doesn't always get a straightforward answer. My name is Ethan Polliner, co-founder of the nutrient analytics platform Cultra. And in this video, we're going to be walking through what biological inputs actually are, how each category works, how to integrate them into a nutrient management program, and how to read a product label with enough context to make a confident decision. Let's get into it. Before we go any further, we need to establish a shared vocabulary, because this is where a lot of the complexity begins. The term biological inputs, or just biologicals, is an umbrella term for a broad category of agricultural products derived from natural sources: bacteria, fungi, plant extracts, minerals, and other organic materials. They're distinct from synthetic inputs, which are manufactured through chemical processes. Now, within that umbrella, there are three main subcategories: biostimulants, biofertilizers, and biopesticides. Now, these categories overlap more than the labels suggest, and a single product can often serve more than one function. The distinctions are still useful though, so let's walk through each one. Biostimulants are any substance or microorganism applied to the plant or the surrounding soil that supports better plant growth without directly supplying nutrients and without acting as a pesticide. They work by influencing the plant's own internal processes, improving nutrient uptake, efficiency, stress tolerance, and root system development. Common examples include seaweed extracts, humic acids, fulvic acids, protein hydrolysates, and plant growth promoting bacteria. Importantly, biostimulants are not a replacement for a fertility program. They help the plant make better use of the nutrients already available, whether from your nutrient management program or from the soil itself. Biofertilizers are products containing living microorganisms that, when introduced to the seed, plant, or soil, actively improve the plant's access to nutrients. The classic example is rhizobium bacteria in a soybean inoculant, which form a relationship with legume roots and convert atmospheric nitrogen into plant-available forms. Another well-known group is mycorrhizal fungi, which extend the plant's effective root zone, improving access to phosphorus and water and other nutrients. Now, biofertilizers act specifically by increasing nutrient uptake. Biopesticides are biological products used to suppress or control pests, pathogens, and disease through living organisms or naturally derived compounds. Some directly inhibit or kill target pathogens, others trigger the plant's own defense systems through induced systematic response, priming the plant to respond more effectively when it actually encounters a threat. And then some act as competitors, occupying space and resources so that the harmful organisms have less opportunity to actually establish. Now, many biopesticide organisms also support soil health and plant growth simultaneously, which is part of why the line between biopesticide and biostimulant can actually blur at the product level. Now that we have that shared vocabulary in place, let's go a level deeper, because understanding what a product is called is one thing, but understanding the mechanism behind it is what helps you make better decisions about when and how to use it. And for a lot of growers and agronomists, this is the part of the conversation that's often missing. Humic and fulvic acids are organic molecules derived from long-term decomposition of plant and animal matter. They're closely related, but behave differently in the soil. Humic acids are large molecules that improve soil structure over time, form complexes with nutrients that keep them in more plant-available forms, and some research suggests they even influence gene expression in roots in ways that support more vigorous development. Fulvic acids are smaller and more soluble, which means they move through the soil and into the plant more readily. They're particularly effective at chelating micronutrients, binding them in a way that keeps them plant available rather than locked up, and are thought to enhance cell membrane permeability in ways that support nutrient and water uptake. Many commercial products contain both, and the two are considered complementary. As a general rule, humic acids tend to have a stronger influence on soil health and structure, while fulvic acids tend to have a more direct effect on plant nutrient uptake and stress response. Seaweed extracts are rich in cytokinins, among other bioactive compounds, including auxins and polysaccharides, which collectively influence plant hormone balance and stress response. Cytokinins in particular are plant hormones involved in cell division and stress signaling. Under heat or drought stress, cytokinin levels naturally decline, contributing to yield loss. Seaweed extract applications can help sustain those hormone levels and support more normal plant physiology under pressure. Plant growth promoting rhizobacteria live in and around plant roots and produce compounds that directly benefit the plant. Some produce auxins, a hormone that drives root elongation. Others produce enzymes that release nutrients from the soil compounds. Others compete with and displace harmful microbes. Nitrogen-fixing organisms like rhizobium colonize nodules on legume roots and convert atmospheric nitrogen into ammonium, a plant-available form. In a well-inoculated legume crop, a meaningful portion of the plant's nitrogen requirements can be met through this process, though the actual contribution varies depending on inoculation quality, soil conditions, and the growing environment. Mycorrhizal fungi extend thread-like structures called hyphae far beyond the physical reach of plant roots, dramatically increasing the surface area for nutrient absorption and a genuine two-way exchange with the plant. And phosphate-solubilizing bacteria produce organic acids that release locked-up phosphorus into the soil solution where the plants can actually take it up. Now, plant diseases are a significant driver of why biopesticides matter. The Food and Agriculture Organization estimates that plant diseases account for roughly 14% of food loss globally, with fungal pathogens responsible for 42% of those losses and bacterial pathogens accounting for another 27%. Biocontrol agents are increasingly becoming incorporated into integrated pest management programs because they can address this problem through multiple mechanisms at once, while also supporting plant growth and soil health, not just suppressing the target pest. Now, it helps that biocontrol agents don't rely on a single mode of action the way chemistry does. They typically employ several direct and indirect mechanisms simultaneously, which is one of their structural advantages when it comes to resistance management. On the direct side, antibiosis is one of the more well-documented mechanisms. Certain organisms produce toxic secondary metabolites that inhibit pathogen growth or kill them outright. Some engage in mycoparasitism, where one organism directly parasitizes another, colonizing it for nutrients. Others work through competitive exclusion, outcompeting pathogens for limited resources and ecological space. And some produce hydrolytic enzymes that split structural components of pathogen cell walls, such as chitin and cellulose, effectively breaking down the physical architecture that pathogens rely on. These same enzymes also play a role in facilitating root colonization by the beneficial organism itself. On the indirect side, induced systematic resistance is worth understanding in some depth. When beneficial microbes like Bacillus or Trichoderma colonize plant roots, they trigger a low-level immune response throughout the plant. Plant isn't mounting a full defense yet, but it's primed. When a real pathogen arrives later, the response is faster and more robust. Think of it like conditioning for an athlete. You're not fighting a disease, you're building the capacity to respond to one later down the road. One additional point worth making about timing. Biocontrol agents applied before pathogen establishment are consistently more effective than those applied reactively. Identifying the vulnerable windows in a pathogen's life cycle within the context of your operation and timing applications to those windows can meaningfully improve control outcomes. This is one of the more important practical distinctions between managing biologicals and managing chemistry, where curative options are more readily available. At some point, every grower or agronomist who gets serious about biological inputs runs into the same practical moment. You've got a crop plan in front of you, a fertility program, a chemistry program, seed treatment decisions, and someone is presenting a biological product and making the case that it belongs in that plan. The question isn't whether biologicals work in principle. The question is, where exactly do they fit? For most operations, they fit best not as a replacement for what's already working, but as a complement to it. A biostimulant that supports root architecture and nutrient uptake can help the inputs already within a nutrient management program do more work without changing the program itself. A biopesticide that induces systemic resistance alongside a fungicide program doesn't replace the fungicide, but it can extend the protection window and slow pathogen buildup. In both cases, the nutrient management program stays intact and the biological adds to its effectiveness. It's also worth noting that the largest share of biological product sales comes from operations that also rely heavily on traditional fertility and crop protection inputs, not exclusively from organic producers. Growers across production systems are integrating biologicals because they complement existing programs and improve the return on inputs already representing a significant cost to the grower. Now, compatibility is a practical consideration worth planning for. Some biopesticides can be sensitive to fungicide tank mixes, and some microbial products can be affected by certain fertilizer salt concentrations or pH levels. Checking manufacturer compatibility data before building a tank mix is always good practice, and a jar test in the field is always a reliable verification step. Now, it's worth addressing the real challenges around biological adoption directly, because they're legitimate and understanding them leads to better decisions. The most commonly cited barrier is inconsistent performance. A biological is a living system, or derived from one, and its performance can be influenced by soil temperature, moisture, organic matter levels, storage and handling history, and the existing microbial community in the soil. If you've tried a biological product and seen strong results in one field and underwhelming results in another, that experience is not unusual, and it doesn't necessarily mean the product failed. It more often means that the biological context was different, and that context matters much more with these products than most other inputs. This is a characteristic of working with biology rather than a categorical limitation, but it does raise the standard of evidence worth applying when evaluating these products. Replicated trial data from relevant environments with transparent reporting of conditions and variability is the correct benchmark. Now, cost is the second barrier. Biological products frequently carry a much higher per acre cost than many other inputs, and in tighter margin environments, they're typically among the first line items to be reconsidered. The case for keeping them in the program rests on return of investment supported by solid data specific to the product, the crop, and the environment. The practical starting point for building that data on your own operation is a replicated on-farm trial with an untreated check strip using the same trial design principles we'll cover in the next segment. A single season of well-structured on-farm data from your own fields is often more useful than a stack of trials conducted elsewhere. Now, the third challenge is the regulatory and labeling landscape. In the US, whether a product is classified as a biostimulant, biopesticide, or soil amendment affects which regulatory pathway it follows and what claims can legally appear on the label. Products registered as biopesticides through the Environmental Protection Agency have completed a more formal efficacy and safety review than products sold under fertilizer law as biostimulants. And that registration process also includes an evaluation of environmental safety and non-target effects. Globally, frameworks vary, with the European Union developing a more unified approach, but no single international standard is yet in place. Now, understanding that landscape helps put label language in the right context, which brings us directly to how to read that label. The label on a biological product is one of the most useful evaluation tools available, and knowing how to read it makes a meaningful difference in product selection. The challenges we just covered around trust, evidence, and regulatory variability all have practical answers that start right there on the label. In the US, biopesticides registered through the Environmental Protection Agency under the Federal Insecticide, Fungicide, and Rodenticide Act have gone through a formal review of safety and efficacy, including an assessment of environmental safety and effects on non-target organisms. An EPA registration number on a label indicates that the product has cleared the bar, and practically speaking, it means you're working with a product that has been held to a defined and documented standard. Now, that's a meaningful starting point for evaluation. Biofertilizers and most biostimulants are currently registered at the state level under fertilizer or soil amendment laws, though the picture is more complicated than a clean two-pathway system. Some biostimulant products can trigger federal regulation under pesticide law depending on how their label claims are worded, and the boundary between a state-regulated biostimulant and a federally regulated plant growth regulator is still being actively debated at the legislative level. A federal framework specifically for biostimulants has been proposed in Congress multiple times, but has not yet been enacted. Now, for practical purposes, this means the regulatory rigor behind a biostimulant label can vary considerably depending on the product, the state, and the specific claims being made. The practical implication for you as a buyer is straightforward. An EPA registration number tells you something meaningful about what a product has been through. The absence of one doesn't mean a product doesn't work, but it does mean the burden of evidence shifts more heavily into the trial data you can find independently. For microbial products, the active organism listing should identify the specific genus and species and include a viable cell or colony forming unit, CFU, count. The concentration matters more than most buyers realize. Two products listing the same organism at the same price point can differ substantially in viable count, and that difference has direct implications for whether enough organisms survive storage, handling, and application to actually establish a meaningful population in the rhizosphere. Now, as a general benchmark, Microbial inoculant products should ideally maintain viable organism stability for somewhere between 6 and 12 months from the date of manufacture. For non-microbial biostimulants like humic acids, fulvic acids, or seaweed extracts, look for a quantified concentration of the active fraction rather than a general ingredient listing. A product that lists humic acid as an ingredient without specifying the percentage of humic acid in the formulation is giving you less information than you need. So where possible, ask for it. Label claim language is worth reading carefully and with some skepticism as well. A label citing replicated trial data with specific yield or quality results is providing a different level of evidence than one using language like supports plant health or may improve nutrient uptake. The softer phrasing often reflects the limits of what's legally claimable under fertilizer law rather than the strength of the actual science. When label language is general, don't stop there. Request or locate the supporting trial data independently, and when you review it, look for 3 things: replication across multiple locations and seasons, relevance to your geography and crop system, and the presence of a proper untreated control. The last point is critical. A trial without an untreated check strip doesn't tell you what the product actually contributed. It only tells you what the crop did in response. Consistency of formulation is a dimension that does not get enough attention. Some of the more established biostimulant categories, like seaweed extracts, have well-standardized extraction and formulation processes that produce consistent active fractions from batch to batch. Newer categories, including many plant extract-based products, don't always have that same level of standardization, which means what's in the bottle can vary meaningfully from lot to lot. Now, when evaluating a less established product type, ask the manufacturer directly about quality control protocols and whether batch testing data is available. A company that doesn't attempt to answer that question is clearly telling you something about that product. Additionally, storage and handling requirements deserve attention for microbial products specifically, and they're easy to overlook. Viability can be reduced by heat, ultraviolet exposure, or age beyond the stated shelf life, sometimes without any visible change to the product. A biofertilizer inoculant that's been sitting in a hot warehouse or left in the cab of a truck may look and smell completely normal, while the viable organism count has dropped significantly. Check storage temperature requirements, confirm the shelf life, and make sure your handling practices from the time of purchase through application protect the product's integrity. For mycorrhizal products in particular, strain selection matters beyond just the organism listing. Different strains have specific environmental requirements for optimal performance, and the effectiveness of a mycorrhizal inoculant depends on matching the strain to the host plant species, the local soil conditions, and the native microbial community already present. A label that lists only the genus without strain-level specificity, or that doesn't provide guidance on which crops and environments the product is suited for, is a flag worth following up on before committing to a purchase. Now, a quick note on application rates. Biological products don't follow the same rate-response relationship as many other inputs. Many function by establishing populations or triggering biological processes rather than delivering a quantified chemical, which means simply applying more doesn't work the way it might with a fertilizer or pesticide. The labeled rate exists as a research-grounded starting point, and exceeding it does not reliably improve outcomes, and in some cases it can actually be counterproductive. Research consistently shows diminishing returns at higher application frequencies, a pattern likely driven by changes in uptake and assimilation rates across different plant growth stages. The more important variable is timing. A product applied at the right growth stage, under the right soil conditions, will generally outperform the same product applied at a higher rate at the wrong time. When in doubt, follow the labeled rate and optimize timing before you consider adjusting dose. The biological inputs market is growing in response to converging pressures that are not going away. Regulatory scrutiny on certain synthetic pesticides is increasing, input costs have made nutrient use efficiency a financial priority, soil health is increasingly understood as a direct driver of long-term productivity, and advances in genomics, microbiology, and precision application are making biological products more consistent and effective than they were even a few years ago. The biostimulant segment alone is projected to nearly double by 2030, reflecting a niche category moving into the mainstream. The opportunity that tends to get underemphasized is this. The most significant near-term value in biologicals is not replacing what's already in your nutrient management program. It's in using biological inputs to help your program perform closer to its potential. Integrating these tools thoughtfully, based on solid data, and matched to the right timing and context is where the returns are most consistently found. In summary, biological inputs are a broad category of products derived from natural sources that work with your crop's biology rather than substituting for it. Biostimulants like humic acids, fulvic acids, and seaweed extracts improve how efficiently a plant uses the nutrients already available to it. Biofertilizers like rhizobium inoculants and mycorrhizal fungi actively improve the plant's access to nitrogen, phosphorus, and other nutrients through living microbial relationships. And biopesticides like bacillus and trichoderma-based products suppress pathogens and prime the plant's own defense systems, often while supporting soil health and plant growth at the same time. What these three categories share is that they work with biological systems rather than around them. And that means their performance is context dependent, their labels require a more careful read-through, and their integration into a nutrient management plan is most successful when it's grounded in solid data and matched to the right timing and conditions. I'll be honest with you, the world of biologicals can still feel like a lot to navigate. The terminology is layered, the product claims vary widely in quality, and the research is still catching up to the pace of the market. But that's exactly why building a clear foundation in what these products are, how they work, and how to evaluate them matters as much as it does right now. The biological input space is one of the most dynamic and rapidly developing areas in modern agronomy, and we're still in the early chapters of understanding how to get the most out of these tools. The science is deepening, the product landscape is maturing, and the practical on-farm knowledge is building season by season. We'll be covering the biologicals market further as it develops, going deeper on each of the categories we touched on today and bringing you the real-world results as they happen. I'm Ethan Polliner, co-founder of Cultra, an agronomy data platform built to help growers and agronomists make more informed nutrient management decisions. Thanks for spending the time with us today. We'll see you in the next one.